Welcome to Seeking Alpha's CEO Interviews. Quality of leadership is a decisive factor in stock performance, so we provide in-depth interviews with the best and brightest CEOs in the public markets. We publish limited excerpts from our interviews on social media platforms and the full interviews at seekingalpha.com and in the highly rated Seeking Alpha mobile app. To find the full interviews, open seekingalpha.com or the Seeking Alpha mobile app and search for the phrase CEO Interviews, or simply type a stock ticker into the search box.
I just wanted to thank you for joining CEO Interviews. I appreciate you taking the time to speak with us. And I know you have an extensive background. I could actually, going through some of your history, I feel like I could do a separate interview just on your background. But I think for some of our viewers, it would be great to just kind of get an overview of maybe your even your scientific background and just your proficiencies and kind of where you're at today.
Yeah, sure. Happy to kind of chime in. I mean, part of my background that, is extensive. It comes because, of age. You've been at it for a long time.
Right.
But yeah, going back into way back, I did my first degree in sciences and I majored in chemistry. So I took a shine actually to chemistry. And once I finished my first degree, I thought, what do I do with a chemistry degree? Do I go, to a graduate program, or what do I do? But I thought it would be interesting to go into drug development. And so the first thing I want to do is to get an idea about, pharmaceuticals. So I went into pharmacy, took an undergraduate degree in pharmacy just so I could learn about the drugs and learn about their clinical use and whatnot. Then I worked a year in a hospital just to get some clinical experience, applying that knowledge I picked up through the undergraduate program. And then I went back to school and I did a PharmD to really kind of formalize my clinical training. But one thing I noticed with the clinical training that really actually sort of jazzed me was research. And the more I started thinking about it, the more I realized I probably have to do, or I wanted to do, a graduate program or a PhD in pharmaceutical sciences so that I could really, really dig into pharmaceutics. And in my case, I specialized in pharmacokinetics, and then married that, later on with pharmacodynamics to be able to really understand when you take a drug, what does the body do to the drug, the pharmacokinetic piece, and then the pharmacodynamic piece, which is what does the drug do to the body, and to put those two pieces together. So then I became an academic. My first 10 years I spent as a professor at the University of Alberta in Canada. And I was teaching, courses in pharmaceutics, pharmacokinetics, therapeutics, medicinal chemistry. So there was a lot of background that I was using. And I had a research program at the time, which was looking at pharmacokinetics and dynamics of drugs used in cardiovascular medicine. So looking at calcium channel blockers, beta blockers, things like that to lower blood pressure, but also looking at, how to treat things like, abnormal lipid profile. But as I got more and more into it, companies started asking me to consult, how I would try to position a drug so that the drug might have an extended patent life, or, how could they make or modify their existing pipeline to make new drugs. So it occurred to me way back that you could use a lot of different, I would say, novel approaches to creating new drugs. One approach back in the, I would say, 80s was looking at drugs that came out as stereoisomers. So, the left and right-handed form of a molecule. So, for example, maybe it's the left-handed form that has the effect, and maybe the right-handed version of the same molecule has little to no effect. I mean, you never know, right? So it really depends on what drug you're talking about. But then it also occurred to me that in addition to the stereoisomers that we talked about, there's also isotopes. So you can use heavier versions of things like hydrogen or carbon, nitrogen, oxygen, all these different things. So you're not really limited to, let's say, a piece of paper that looks like, your periodic table,
Right.
But you can start building up molecules that way. So, I thought I had a lot of ideas and I started patenting some of these ideas. And that led to my first drug that I started working on, which was a drug I discovered in the 1990s, which was a modified version of cyclosporin. So I knew that cyclosporin had a lot of side effects, really damaging to the kidneys. And it was what we call a narrow therapeutic window or narrow therapeutic index. So how to make I would call it, new improved version of cyclosporin. And to do that, I thought I'm going to have to leave the university because I need to choose, which horse I want to ride on. So to me, it was, I hate to maybe say it, but it was a no-brainer. I really wanted to get into industry. So I started my first company in June of 1993 to pursue development of the new improved cyclosporin. And I'm glad or happy to say that in January of this year, so, quite a few years later, 28 years later, that drug I started working on way back in the mid 1990s got approved by the FDA. So that's a drug called voclosporin,
Right.
which is an immunosuppressive drug. So it suppresses the immune system, and it's used to treat, in this instance, lupus nephritis, which is a pretty severe kidney disease. So that's kind of the evolution of, I would say the quick summary or a summary of how I got to be where I am today.
Okay. And that's fantastic. And I was actually going to, I had the genesis of CRV 431. I was diving into isotechnica and your background there a little bit. And it was about 10 years that you were almost there developing that out. It's crazy to see how it took now for that to be approved. So the process is lengthy. Absolutely.
Yeah.
And I appreciate the background too. And I think, it's such a complex topic, right? Getting into, receptor sites, this can go really deep. I think for the audience, in terms of when we look at like NASH versus NAFLD,
Yeah.
if you could just contrast like the differences between the two, like is one more treatable than the other? Is CRV specific to NASH? Could you talk about the difference a little bit?
Sure. So NAFLD is non-alcoholic fatty liver disease, just an acronym that makes it much easier for us to talk about it.
Right.
NASH is another acronym that stands for non-alcoholic steatohepatitis. And really, NAFLD or the fatty liver, I call it, is quite common. Probably there's about 25% or so of the global population that has a fatty liver. So if you think about it, you would almost assume that obese people or, morbidly obese people, in fact, could have a fatty liver, but that's not necessarily the case. You can have actually fairly average looking people that have a fatty liver, and there's something even called lean NASH. So it's a more severe form of fatty liver. But to put it in context, 1 in 4 people globally has a fatty liver. Interestingly, and maybe not so good, most people have no idea that they have a fatty liver until maybe it, in a way, it gets too late. And what I mean by that is that if you leave the fatty liver essentially untreated or under controlled, you can develop inflammation of the liver. You can develop even a sort of a scarring of the liver that we would call fibrosis of the liver. And so you start to lose liver function, and the downstream consequences of all of that is potentially to progress to liver cirrhosis, and also perhaps the need for a liver transplant. So NASH is now becoming, leading cause of liver transplants. If you leave that, not well controlled or mismanaged or not controlled at all, it can lead to liver cancer. Most often we talk about hepatocellular carcinoma, which we also call HCC. And, that's a real tough one to treat. And again, it can lead to death. So the clinical sequence of events can be quite dramatic. So the way I categorize it, in my mind at least, is a fatty liver is one thing, but the NASH is a more severe form of the disease where now you've got inflammation, you've got fibrosis. And so what our company Hepion is trying to do with our lead asset, which is called CRV 431 or just CRV, I'll call it, it's trying to address the fibrosis, because the thinking is that fibrosis, once it's the collagen fibers, which is a protein that's laid down to produce that scarring. Once that happens, it's almost too late to try to reverse that really nasty fibrosis. But at least, that's some of the thinking that's out there. But I'm not necessarily sure that that's really the case. There's this push and pull. It's an active process of laying down the fibrosis of collagen and sort of the ripping apart of the collagen. So it's an active,
Right.
push and pull, I would say. So what we're trying to do is we're trying to tilt, the floor, so to speak, so that you're actually kind of reversing the fibrosis that occurs in it. It's a tall order, but I think if we can get there, which we think we can, then I think CRV could become a pretty hot drug in the treatment of NASH, which again is the more severe form of the fatty liver disease.
Right. And I love what you said there, that, physical appearances are not always an indicator of potentially having a fatty liver. So you could see physically a very skinny person, but it's not. And I was going to ask, is it binary? Like if you see a person who potentially, okay, they are morbidly obese, yes, they have a fatty liver, but it's not always one-to-one is what you're saying.
Yeah, I would say so. I mean, there's, the incidence is going to go up, obviously, depend on lifestyle and your diet, things like that, exercise. So we know that one of the best treatments right now for fatty liver is just more exercise, better diet, all those types of things. But we all know that, even from personal experience, that come January and New Year's Day, you're okay. I'm gonna, for sure, this year is going to be different than last year. I'm going to get on the treadmill. I'm going to eat properly. Eat my vegetables and all the mommies tell us, right?
Right.
And then, but you tend to just reverse. You just tend to fall back in your old habits, and you regress. And so that's kind of the nature of the beast, so to speak. But the trick from a clinical perspective is that how can you diagnose these people with, for example, a fatty liver, so that you can, ward off, or you can cut off the progression of the disease before it actually progresses to NASH. Like, how do you do that? Because if you don't know that you've got a fatty liver, and you're one of those 1 in 4 people globally that has a fatty liver, and you don't know it, because in a way it's like hypertension or high blood pressure, where you have that silent killer, so to speak. Like, if you don't know you have it, why would you go to the physician's office and say, I think I have a fatty liver? And there's no simple blood test. There's no simple, really easy way to diagnose it. That's the trick. And so by the time you do realize you might have a fatty liver, again, in a way, it's a little bit late. And so you're chasing your tail, so to speak. And by then you probably do have a fair amount of inflammation and fibrosis. And that's where we think we can really make a difference, because most of the drugs in development right now in the NASH space are drugs that are addressing the fatty liver. In other words, the events leading up to NASH. But again,
Right.
if you don't know you have a fatty liver, why would you take a drug that you don't know you even need? So it's
Right. How
confusing.
do I know my hepatocytes are under stress? How do I know? Is there a way to know, aside from like a biopsy, or just if I have a reduced functioning liver, what are some of the ways someone might know that, where they may need maybe Hepion's product could benefit them? Is there a way that someone could know, just kind of as a layman, or is it really hard to tell?
I think from a clinical perspective, if you're feeling lethargic, you're feeling kind of down and, not quite right. And maybe even you've got some changes to your skin color and tone, those types of things.
Right.
You might hopefully you go into the physician's office before then, but you'll see a rise in what we call these liver function tests, or things like the transaminases. And specifically, we look at ALT,
Mhm.
which is one of the transaminases. It's an alanine transaminase, and then an AST, which is for aspartate. But I shouldn't say either, both your ALT and your AST will rise. And what that's telling you is it's a very non-specific type of test. It'll go up for a lot of different reasons, but that's one of probably the first things that you'll see is liver enzymes look a little bit off kilter.
Uh,
So there might need to be more digging, so you start looking a little bit further, and then eventually The only real way to so far, to get a good handle on this is to do a liver biopsy. Nobody's going to, look forward to a liver biopsy, I don't think. So again, there's diagnostic companies out there that are trying to get a really good grip on what does it take to diagnose something like NASH, and how, even after diagnosis, if you treat them, how do you monitor the improvement, I would say, or not, in the clinic by taking these non-invasive types of markers. And in other words, how do we avoid liver biopsies? But that being what it is, liver biopsy is the go-to eventually, and it's still I would call it standard of care, in terms of diagnostics.
Right. And when you talk about like the release of inflammatory molecules from the death of hepatocytic cells, you guys were able to pinpoint down on cyclophilins, but there's also like cytokines and other inflammatory molecules that are released, right? Was there a reason that you chose cyclophilins to zoom in on as really, or are they weighted differently? Or did you guys choose cyclophilin for a specific reason to kind of focus on that?
I think first, yeah, it's like which came first, the chicken or the egg.
Right.
So in our case, we've been working on cyclophilins for, well, in my case, since about the late 80s. And most of our group, in the R&D group in Hepion, has been working similarly that long on cyclophilins. Again, our first drug, voclosporin, it actually binds to cyclophilin first of all. And we do know that in the human, there's 17 different, we call them isoforms, but 17 different types of cyclophilins. And, cyclosporin and our last drug, voclosporin, all of those things will bind to cyclophilin, same with CRV431. But the difference is between voclosporin or cyclosporin is that they bind first of all to cyclophilin. So that's kind of, if you want to call it, the dance partner.
Right.
But then that dance partner is looking for a third partner. In this case of voclosporin and cyclosporin, they look for another thing called calcineurin. And by binding calcineurin, that's where you start kicking off the immune response, or immune suppression, I should say. And that's why drugs like voclosporin and cyclosporin can be used for solid organ transplants, for example, kidney transplants.
Right.
They can also be used to treat autoimmune disease where your body kind of loses control of its own mind in a way. I'm just saying that from immune response standpoint.
Right.
It can't recognize your body as self. It thinks certain parts of your body may be foreign. So it starts to reject, let's say, skin, in the case of psoriasis or lupus nephritis or lupus. It doesn't realize that those things that are in your body actually belong there. So it starts rejecting it. In the case of CRV, we took away the binding to calcineurin altogether. So there is no calcineurin binding. In other words, there's no immunosuppression.
Right.
So what we did is we focused on the binding to the cyclophilins. And then, for example, if we drill down a little bit into amongst those 17 different cyclophilins, we started looking at specifically three. We looked at cyclophilin A, which is in the cytosol of the cell. Cyclophilin B is we looked in and we find that in other parts of the cell, and we do notice that cyclophilin B plays a big role when it comes to the production or the synthesis of collagen, which is, of course, the collagen is the protein that causes this fibrotic scarring.
Right.
Another one called cyclophilin D, which is resides in the mitochondria, these little energy power plants in our cells. And so by inhibiting cyclophilin D, we think we can protect cells from injury and death ultimately. So there's all these different roles for cyclophilins. So once we studied the cyclophilins and we knew what was going on with cyclophilins, we said, okay, what diseases could we potentially target knowing all of this information about cyclophilins? So then we said, okay, well, we can use cyclophilins for sure, probably in antiviral treatment.
Right.
So we looked at hepatitis C, we looked at hepatitis B, we looked at HIV. We also looked at hepatitis D or Delta virus. Even recently, we looked at coronavirus 2 because we know that coronavirus also needs cyclophilins. Mm-hmm. But then we looked at, what other potential ways can we use cyclophilins or antagonism of cyclophilins to our benefit to create drugs? And then again, we found liver disease. For example, you do have the fibrosis, therefore you have a role of cyclophilin B. You also have inflammation, so there's a role for cyclophilin A. And ultimately, when you start getting cell death, like your liver starts, to be injured and die, the cells in your liver, then there's a role for cyclophilin D. So it seemed perfectly suited to treat liver disease. The one other piece I'll add to that is that we do know that the chemical structure of cyclophilins or cyclosporins and drugs that are related to cyclosporins, after you take that drug orally and it's absorbed through the GI tract, they target the liver. So it's almost like the liver is a sponge, and it's the first thing the drug really sees when it enters, I would say your systemic circulation, is it slams into the liver and it gets stuck there. It's what we call hepatic extraction, and it's highly extracted by the liver or hepatically. And that's why we decided, okay, well, let's target liver disease, because even though there's fibrosis that could occur in any number of organs, could occur in the heart, the lung, the kidneys, even on your skin for that matter. When you cut yourself with with your kitchen knife, you get a scar, right? But when the drug is zeroing in on the liver, it seems like, well, that's where you should take your drug, and that's where you should, chase after an indication in liver disease. So that's why we landed on NASH, and so far it's worked well.
Absolutely. And the video on your website is fantastic. It actually gives a really good breakdown of even like mitochondrial stress, like pore leakage, in the process of how that's really, I don't want to say closed up, it's not the technical term, but how that process reverses with CRV 431. So very interesting.
Yeah.
And just another just general liver question. So can the liver heal itself? So if you have cells that die, or you have CR431 that's introduced to the body, are you starting kind of from ground zero? Like, let's say you have 50% reduced liver capacity after the drug. Can you have an increased liver capacity, or are you working from a new baseline? Almost like if someone had like alcoholic disease where they had to have part of their liver removed. I've heard stories of, I was able to survive with a tiny liver left, just a little bit, but I can't drink. Is that, I don't know if I'm forming this question right, but does that kind of make sense? Like, do you still have function? Like, do you have renewed functionality of your liver? Have you seen that in any models at all, or anything like that?
We haven't. Yeah, we haven't extensively studied that. We haven't specifically studied that either in the context of our drug development of CRV. I mean, livers are known to regenerate, and that's why when you do liver transplants, you don't have to take the whole thing. You can put in a lobe, and it'll start to regenerate. So there is that aspect, and even in the context right now, with your question, I was thinking of drug development. There was a recent drug that I believe it was approved in Europe called Mercludix B for the treatment of hepatitis B. And what Mercludix B does is it inhibits the uptake of the hepatitis B virus into the liver cell, the hepatic cells. And if you think about it, well, if you've already got hepatitis B, how effective would a drug like that be? But we do know that cells turn over, so if a cell turns over and it's got the virus, if somehow that cell was able to turn over and maybe potentially the viral load could come down, if you're blocking the virus from going in again, or recycling or whatever it does, maybe in theory and in reality, again, the drug got approved. So maybe we can start treating things like hepatitis B because of that ability of these cells to turn over. And so if basically you put it this way, you have a house party, which we can't do in the days of COVID. Maybe it's getting better and better.
Right.
But imagine you've got a house party, you've got 50 or 100 people in your house. And of the 100 people in your house, let's say you've got 50 really obnoxious guests. And, you just want to get rid of them. So you can open a door and you can, you can toss them out basically. And if you don't want them to go from your back door to the front door, lock the front door. So maybe that's in a way like how you block the uptake through drugs like Mercludix B. So you're recycling, or you're, in this case, pushing things out and then locking, the uptake. And that's basically what you're trying to do. So again, that's maybe a
Right.
round way of talking about regeneration, but
Mhm.
eventually, once you get rid of the obnoxious gas, you've got a perfectly functioning liver. How's that for an answer?
No, it's great. It's a brilliant analogy, and this is a physical turning over of the cell, is what you're referring to, right? Where there's just more surface area for potentialities, basically. I think that's a great analogy.
And we talk about things like half-life, right? We
Right.
know that, for example, red blood cells last about 120 days. So if you have a problem with a red cell, if you're able to fix the problem, maybe 120 days later, things start getting better. So we're using the body's natural ability to turn over things, and we're intervening to try to, in a way, shut the door. Or if you can't shut the door, at least you can try and, open the back door and kick people out, or in this
Right.
case, the bad things from happening. So that's
Mhm.
kind of, I know it's a simplistic way, but it is
Perfect.
like talking about turnover. Maybe that's a kineticist in me talking,
Right.
because we talk about half-life all the time.
Right. And I think that's why 3D models are becoming really popular now. I think people want to work in a lot of 3D, at least from a cell culture standpoint, because you have more, it's more representative of what's actually going on in the body. So I totally get that. Um, I had a question regarding Hepion CRV 431. It's a modification of vocalosporin. I don't want to understate that, but it seems like there's like different functional groups being added to the molecule. And I think you mentioned a 13-fold increase in affinity for cyclophilin. Is that proprietary, or is that something that you could talk about, that 13-fold increase in going from cyclosporin A to vocalosporin to CRB 431, if that makes sense?
Yeah. So CRV 431 is a chemical derivative, I'll call it, of cyclosporin. Not really voclosporin, but cyclosporin. We use the voclosporin, knowledge that we gained over about 25 years or so working on that in the field, to be able to come up with, molecules like CRV 431. So we start with cyclosporin, as that is our starting material.
Right.
And then what we did in the case of voclosporin is we modified amino acid 1, I'll call it. So really what cyclosporin is, it's a series, like it's a string of amino acids, and the string is 11 amino acids long, and it's hooked, into a circle. So it's a ring structure of 11 amino acids. And by convention, chemists have numbered these 1 through 11. So we made a modification on amino acid 1 in a way that made voclosporin, I would say, much easier to use clinically. When we started working on pure cyclophilin inhibitors like CRV 431, we went back to the drawing board. We started again with cyclosporin. And in this case, what we wanted to do is we wanted to get rid of immunosuppression entirely. So we made, in this case, 2 chemical modifications, one to enhance the binding of cyclophilins. That was one modification. The second modification was to obliterate the immunosuppression quality of the cyclosporins, so that we could target purely cyclophilins. Then what we did is we had to go to the lab to test, what is the binding affinity of CRV 431 against, cyclophilins, and how does that compare to things like voclosporin and cyclosporin. And even we knew at the time Novartis was developing a molecule called alisporivir. So we also bought some of that. We said, well, how does our binding affinity compare to alisporivir? We haven't found any cyclosporin analog or chemical derivative that binds so so cleanly and so tightly with cyclophilins as CRV 431. It's a real strong binder. It's got a high affinity, high avidity type of binding.
Mhm.
I think that's why it's probably a fairly potent molecule in certain disease, the treatment of certain diseases. So that's kind of like
Right.
a screening assay we did at the beginning.
Okay, I can only imagine when the mechanism was discovered, maybe the excitement in the lab. I can only imagine the amount of cell culture work that's involved in, your team doing this. I mean, the trial and error is probably extensive, right? I mean, I can only imagine
Oh
it's
yeah, huge. And so the guy who did all that for us is Darren Year. Darren is our chief scientific officer, and Darren joined us when we were way back at Isotechnica. Darren joined us in 2003. He had just finished a postdoc at the Mayo Clinic in Rochester. So we recruited him up to Isotechnica, and he became, the molecular biologist that was able to do all that. So he joined us in 2003, and he's currently working at Hepion. Another fellow, Dan Trepanier, he does all our formulation work. He does our toxicology, he does our patents, all that. Sort of a jack of all trades, but he's an expert in all of them as well.
right.
And he joined us in 1997. So we've worked together, what is that now, about 25 years or so? No, yeah, 25 years at least. And then another fellow who's works in our clinical pharmacology group. He does all our AI work. I've known this guy since 1976, and we worked together going back into the 80s. So, our team, our R&D team, like, they're like me. They're all, long in the tooth and lots of gray hair. They're hopefully they don't watch this interview.
Well, that's a good thing, right? They always say you want a experienced surgeon, but a not experienced barber or something. I forget the metaphor exactly, but I think it's over 100 years of combined experience. So it's amazing.
Yeah, exactly. With the four of us, we're probably well over 100 years of experience. And, it used to be that companies like Sandoz or Novartis, the company that developed cyclosporin originally, they had the world's expertise on cyclophilins and cyclosporins, but those guys are long gone. And I can confidently say that there's no group that has our level of expertise in cyclophilins globally. I don't think there'd be anybody that could match our experience level now. We've been at it for so long.
Are you also sort of almost like, I don't say layman's, but with ADME and DMPK type studies? I mean, you're probably doing a lot of those models in the lab, I would think, or just assuming that you're using hepatocytes in the lab. Maybe is that kind of part of your as well, because after the binding takes place, does the body see that as a waste now and it has to be removed after it binds to so cyclophilin A, B, or D? After that happens, is that now just in the body as just waste, or like what is that process like, or that mechanism from an ADME standpoint?
Yeah, so from an ADME perspective, if you break it down, ADME is absorption, distribution, metabolism, and excretion. And so each one of those four processes are things that we study. And of course, I'm a kineticist, a pharmacokineticist by training. And, Pat Mayo, I just mentioned, he's been around with us for a long time too. He's also a pharmacokineticist. So we break those down into bits and pieces. So the first piece, of course, absorption, like I mentioned, CRV is perfectly suited to target liver disease because after the drug is absorbed, it goes straight to the liver. Then from a distribution profile, we look at where does the drug go? So for example, if we were to look at, does the drug cross the blood-brain barrier? Well, probably not so much.
Right.
So can we use the drug to treat, for example, diseases that affect, the central nervous system? Maybe not nearly as well as we could treat liver disease, but, we haven't really poked around too much either in that field, to be fair. Metabolism, we want to be able to figure out whether the drug is metabolized in a way that produces these, these kind of waste products that you mentioned.
Right.
And then all of this, of course, you have to take into consideration what we call protein binding. And going back to your question about cyclophilins, cyclophilins are proteins. And so to what extent does that binding elicit a response, or what does the body do to that after it is bound? Is it going, basically into waste? What we do know is that that binding is required to be able to elicit a pharmacologic response, which is why we look at the pharmacodynamics. And ultimately, the excretion of the drug, does it go out, through the liver? Yes, in the case of a drug like ours, probably it finds most of its way out through the liver, secreted through bile, things like that. Whether drug, that's intact drug or whether it's metabolized drug. And then how much does the drug either get out through the kidneys or maybe even exhaled metabolites? So this probably not a lot coming out through the lungs because it's not volatile, but those are all the things we have to consider. And those are things that can also affect what we talk about off-target effects. So is your drug safe? That's what the FDA cares about mostly.
Right.
We've studied the living daylights out of this drug to make sure that we're not seeing these off-target effects. So it's not indiscriminately binding to proteins that are going to cause all kinds of problems. We've studied it extensively. This drug is incredibly safe. We're up to probably, I would say, close to 100 people that have taken our drug. We're not getting any adverse effects really to speak of. I remember in our phase 1, we had one person said, well, they developed chapped lips. I mean, I don't even know what that actually means. I do know what chapped lips are, but it's something you have to document. Everything you have to document,
right.
Whether it's related to the drug, probably not. We've also done extensive toxicology studies. We're not seeing any signals. So drug appears incredibly safe. I mean, we, in a way, joke around a little bit and, in amongst our four walls saying, well, maybe we should just put this in a water supply. It's so innocuous, but yet it can do so much because cyclophilins, they're ubiquitous. They're enzymes and, proteins I mentioned, but proteins in this case that are enzymes that control the way proteins are folded. So you can imagine, like you and I sitting here across a Zoom interview right now, everything in our body is protein, like we're proteins, we're carbon units if you watch Star Trek, and we're the proteins have to be folded. So we have a linear sequence of amino acids that can eventually create peptides and proteins, but those have to be folded either, kind of like,
Right.
a three-dimensional structure, and that structure will determine the function of those things. So if you have a disease where you have misfolded proteins, if you throw in a cyclophilin inhibitor that can kind of straighten it out and fix it, you have potentially a drug that could treat an indication. So it's that misfolding of proteins that seems to be at the core of what we're trying to do here.
Right. And that's why I think CRV 431 was so powerful, because you can target multiple diseases with a single agent. I think that's the most powerful thing. And Dr. Foster, we are running up against our time. But if you could just give a brief, like a few minutes, I think you guys are moving into a phase 2. You have a phase 2A study going on now. And we'd even love to maybe talk to you again in the future about maybe the phase 2B and maybe a little bit more about the AI power system that you guys have, which is incredible from an informatics standpoint. Just getting dose responses correct, and it's just incredibly powerful. We could probably talk for an hour about that as well. But yeah.
Don't get me going.
But yeah, if you could just kind of close out with your phase 2A trials, and just we appreciate your time.
Sure. So the phase 2A trial was designed to look at, well, first of all, we did the phase 1, which was looking at our drug in healthy volunteers. So here again, you're looking at pharmacokinetics. You're just trying to study the safety of your drug, and you're trying to characterize the pharmacokinetics in healthy subjects. Now, when you go into phase 2, you're taking actual patients, in this case, NASH patients. But the question is, when you have a NASH patient, and certainly in the case of a liver disease, and you have a drug that's handled by the liver, does that alter the pharmacokinetic profile? Does it alter the way the concentration of the drug changes over time when you have people with livers that are not completely healthy? So we really wanted to characterize that pharmacokinetics in NASH patients. But we figured, okay, if we're in there and we're looking at a 28-day dosing length of CRV at a couple of different doses, while we're there, why wouldn't we take the opportunity to look at any potential biomarker that may give us a glimpse into whether or not CRV could potentially work on a larger scale phase 2, which we call a phase 2B, or even later in the future, a phase 3 trial? Is there anything we can garner from looking at the tea leaves, so to speak, that will tell us whether CRV is going to work? Because these trials are expensive, many tens of millions of dollars. So before you go and spend all that money, make sure as best as you can, your drug will actually work. So to that extent, our phase 2A is looking at PK or pharmacokinetics. It's looking at some of the biomarkers like the ones I mentioned earlier, ALT, AST, and we're trying to also get a sense of, anything else we can look at,
Right.
including bioinformatics. So by the end of the whole process here for phase 2A, we're hoping that we're going to see a good decline, significant decline in the ALT, the AST. That tells us a little bit about the liver health. And we'll look at the other biomarkers. So for that, we'll be doing what we call multi-omic analysis. We'll be looking at genomics. We'll be looking at potentially, things like transcriptomics and lipidomics. So we'll be looking at, lipids, RNA, proteins, genes, those types of things. And that'll help us as we move forward into, the phase 2B and the phase 3. So we're looking forward at getting these trial results out by the end of this quarter. So we're now into the second quarter of 2021. By the end of this quarter, we're looking at top line data. Sometimes, even though we get the top line data, by the time you get database lock and everything else and get your full on report from your CRO, that takes time, but we should get a really good sense of that by the end of the quarter. And then later on this year, we'll go into the phase 2B trial, which is a larger trial. It's going to be a longer period of time. It could probably be about a year of dosing, maybe a quarter, a year or so of follow up with the patients, but that will be much, much more in depth and a lot more patients at a couple of different doses. So we're just kind of putting the finishing touches on putting that together, but we're anticipating starting to screen patients and getting that whole process going towards the end of this year. So it's a big process. And NASH, I have to say, NASH is a little bit, it's a little bit scary. Companies have struggled in NASH because, again, I think we're using a different approach. We're an antifibrotic. We're a once-day oral, direct-acting, hopefully, antifibrotic, as compared to a lot of other companies that are looking at how do we reduce liver fat in the hopes that downstream you may have a reduction in your fibrosis. And companies have struggled because targeting liver fat may not quite be what you need to tip the scales to get enough antifibrotic effect. And it's going to take a long time. So I think that's where some of the challenges are right now in the NASH development space.
Absolutely. And I think your presentation did a fantastic job on the website, kind of detailing placebo effects and things you guys have had to deal, the industry's had to deal with historically.
Right.
Yeah. And Dr. Foster, I think we're running up against our time, but
Okay.
yeah, I just want to thank you very much for joining us. I could talk to you for a long time about all of this. So it's, but you guys have a lot of, Hepion has a lot of exciting stuff coming up as you work through your 2A and 2B trials. And hopefully we could speak again in the future as you move through those and maybe dig into the AI platform a little bit more.
Yeah, we'd love to.